Today's topic is about light effect question. Here you can see the photograph of chloroplast and different structures are being labeled here. So first we are starting with this the membrane of this gram. The membrane of this gram is here. You can see the gram is just stacked over each other. So this gram is made up of many grams which are stacked over each other, and the membrane of the gram. This one is the membrane of the grana, and this one is the membrane of the grana. Remember, these grana are interconnected by lecture. These are the proteinous strands. So this grana is the light harvesting system in which light is being tracked. So here you can see the membrane structure here. This one is the membrane structure. This which is from one grana thylakoid membrane is there in which the photosystem exists. Photosystem is actually the light tracking system or light harvesting cluster in which light is being tracked. So it is like a funnel shape type. Here you can see funnel shape here with one conical like that. So here arrangement of the pigments is like many accessory pigments is surrounding the primary pigment. Primary pigment is here in the center. That consists the chlorophyll A and chlorophyll B. While this accessory pigment which is in surrounding all around circular shape or a conical shape, that is accessory pigment. And the accessory pigments are carotenoid and xanthine. So these are the light harvesting cluster in which light is being trapped. So light is trapped here in the graph, here in the thylakoid membrane, here and here and here. So this is the, you can say, light harvesting system. So the function of these accessory pigments is to transport the light energy into the primary pigments, which is the reaction center actually. Why we are saying reaction center? Let's begin with that. So here is the phospholipids bilayer present here, from here, which is thylakoid membrane here, and then from here. So up to this portion is the thylakoid membrane, which contain phospholipids. Phospholipids are the molecules which are arranged in a bilayer. So this is one, suppose, phospholipid here, right? And the other phospholipids will be on the other side, for example, here. So this is to understand that what is thylakoid membrane, and it is made up of phospholipids bilayer, and certain proteins are being arranged. For example, one of the protein is of this funnel shape. I have told you earlier that photosystem. There are two photosystem 2 here and photosystem 1 here. All right. Within the thylakoid membrane, these are the two photosystems. And each photosystem is the light harvesting system, which consists of primary and accessory pigments, as I have told you earlier. So first, here light is trapped. So light is being trapped here, and this light, which is being trapped by the accessory pigment, focus into the reaction center, I mean the primary pigments, breaks down the water molecules. What's water molecules split up, you can say, that is photolysis, and during which protons, I mean the hydrogen ions, and electrons are being released, along with oxygen gas is being produced. The balanced chemical equation need to write, in which one water molecule can produce one atom of oxygen. So as a whole, it doesn't look nice. So you have to write two water molecules here, then hydrogen ions, will be produced. That is the 4 protons, you can say. 4 electrons will be released from 2 water molecules and 1 oxygen molecules will be produced. So anyway, you can write, if you are writing 1 molecule of water, then 1/2 oxygen molecule you have. If you write 2 water molecules, then 1 oxygen molecules will be released. So 2 mass equations will be there in the exam paper. So it is occurring in the photosystem 2, right? So photosystem is that where light is being trapped over a certain wavelength. And in photosystem 2, the chemical process is photolysis, in which water molecule is split up. The protons which are being released is diffusing here and there in the phospholipids thylakoid membrane and may escape from here into the stroma, which is just outside the thylakoid or crana. While the electrons are moving to the high energy level in this phospholipids layer, so they can also escape from here. But instead there is a system, electron transport chain, you can say, ETC, electron transport chain is here. So in which certain proteins are being arranged in such a way that electrons are moving from high energy level to the low energy level, releasing energy, and this energy will be utilized to diffusion of protons across the ATP synthase. Again, chemiosmosis occur and ATP will be generated. The ATP which is generated here is from ADP and phosphate, so it is so phosphorylation process, but we see it of photophosphorylation. So you can see that it is photophosphorylation because the electrons and protons which are being produced is due to the light which is being trapped here in photosystem 2. And electrons and protons produced from here are moving within this electron transport chain. There are certain proteins among which ferredoxin is one of the most important protein in the arrangement. As a whole, we are saying ETC, I mean electron transport chain, and electrons are moving here and producing ATP energy by movement of hydrogen ion through ATP synthase. So ADP plus phosphate release ATP. So this is photophosphorylation. And it is a non-cyclic way because it is starting from the photosystem 2. And when these electrons are reaching to the photosystem 1, again this photosystem 1 is the system to trap the light here. When light energy is trapped, again the electrons are moving from here, being excited and reach to the highest energy level within the phospholipid and may escape from this. And if they do not escape, they fall back into the ETC because ETC is specified for electron transport. So these electrons moving back into the ETC and again moving through the ETC, they release ATP from ADP and phosphate. So again, chemiosmosis occurs. So in this way, it moves in a cyclic way, reach to the photosystem 1 again, and then again light is absorbed and moving to the high energy level again within the phospholipids, and then again fall back. This is one of the fact that they move in the cyclic way. So that is cyclic photophosphorylation. That ATP is being produced again and again, and this cyclic photophosphorylation moves around the photosystem 1 only. So what is happening in cyclic way of photophosphorylation, that photosystem 1 absorb light energy and electrons which is being present here are moving to the high energy level, which will fall back into the electron transport chain. And moving to the ETC, they cause the movement of hydrogen ions, which result in production of ATP. So this is cyclic. The non-cyclic way is starting from photosystem 1 and then photolysis producing protons and electrons, and these electrons then move to the ETC and then energy is being released, and during which ATP is being synthesized by the movement of protons and then reach to the photosystem 1. And then they can again trap light energy and these electrons can escape through the phospholipids. This is one of the choice that they can escape. So ATP can be produced by cyclic and non-cyclic way. Again, I repeat, the non-cyclic photophosphorylation consists of photosystem 2 and photosystem 1 both. The electrons and protons which are being produced here during photolysis can immediately diffuse out of the phosphoripars bilayer. But if they reach to the high energy level, I mean the electrons, they move through the ETC, which causes the production of ATP by photophosphorylation. In other case, they can escape out. So if we as a whole look at this light dependent reaction, which hold the ATP synthase mean that through which chemiosmosis occur by passing of hydrogen ions, ATP is being generated here. So if we see it, chemiosmosis and light dependent reaction, the total productions which is number one, a lot of ATP is being produced, a lot of ATP molecules by cyclic photophosphorylation or non-cyclic photophosphorylation. Then number two, the production of light dependent reaction we are talking about. Number two is protons and electrons. And you know that the carrier of these protons and electrons is NADP. So NADP is the carrier of protons. So it will become a reduced NADP. NADPH. So it will become reduced, right? So this is the production of light dependent reaction. One more production is the oxygen gas, O2. So this oxygen gas is although the production of light dependent reaction, but it acts as a waste because it is no more used during light independent reaction, which is the second stage of this light photosynthesis. So the things which are being used here, they are ATP, which is required for light independent reactions, and then number two, reduce NADP. These two things will be utilized in the next step of the photosynthesis, that is light independent reaction. So the third product, oxygen, may be used during cellular respiration, I mean in the mitochondria for oxidative phosphorylation. But in daytime, it diffuses out of the stomata and released into the air. So this is as a whole light dependent reaction, which consists of cyclic photophosphorylation here. This one is the cyclic photophosphorylation, and this one is the non-cyclic starting from photosystem 2 and then passing electrons through the ETC and protons passing through the ATP synthase, and then electron reaches the photosystem 1, and then by absorbing light energy they can excite to the high energy level. So this is an escape out from the phosphoripars. So that is a non-cyclic way of photophosphorylation. So the first step of the Kelvin carbon dioxide fixation. Carbon dioxide fixation. During this process, carbon gas which diffuse at and reach in the air spaces is available for the light independent reaction. There is one compound in the stroma of the chloroplast that is known as ribulose bisphosphate. It is a 5-carbon, shortly known as RUBP, and this is one of the most organic compounds present in the living organisms. So this RUBP, which is 5-carbon compound, combined with 1 carbon dioxide plus. So after carbon dioxide fixation, when GP is being produced, GP will be converted into TP. This is the second step, during which ATP is being used and it is hydrolyzed into ADP and phosphate. Energy is produced here, and this energy is used during this reaction to convert G. The second product of the light dependent reaction is reduced NADP. And here reduced NADP is oxidized to release hydrogen ions, which is required for GP to be converted into TP. Two molecules of triose phosphate will be produced from these two compounds, glycerol phosphate and reduced NADP. NADP is set free again, and this NADP is again available in the thyroid membrane to carry more and more protons from there. So in this way, these two products of the light dependent reactions, ATP and reduced NADP, is being used here. The second step of the Calvin cycle, that is the light independent reaction. Then this TP can be converted into glucose, which is the primary product of the photosynthesis. So glucose is being formed. And the second choice for TP is to make RUBP, which is a 5-carbon compound. And the first priority of this TP is to convert into a 5-carbon compound that is 5 times as compared to the glucose. That is 1 time, so 5:1. TP has more chance to be converted into RUBP, so that RUBP should remain in abundance in the stroma to be available for carbon dioxide fixation, because the carbon dioxide concentration increases as the day passes, and the CO2 is available to convert RUBP into GP. So there is a 5:1. Why 5:1? Because RUBP should remain in abundance, and it should not be act as a limiting factor. So anyway, here comes the glucose. The glucose can be converted into further into amino acids when nitrates combine with the glucose in the leaf, and these nitrates are being supplied through the xylem methods, which is being transported from the roots to the leaves, and here they combine with glucose to form amino acid, and the amino acid further form proteins in the leaf or whatever the parts they are being transported through the xylem, through the phloem sieve tube. As in AS transport in plants, we have then these amino acids are being transported through the sieve tube, and so they will be converted into the proteins in the cells and tissues of various organs of the plant. Depending on the type of the tissue and plants, they are converted into hundreds and thousands of different types of proteins. Glucose can be converted into fats molecules also, and the glucose can be converted into further many more organic compounds like cellulose, sucrose, starch, and many other carbohydrates molecules. So when this TP is converted into RUBP, again ATP molecule is being used. So ATP is used here also. So that's why in the light dependent reactions, a lot of ATP is being produced during cyclic and non-cyclic photophosphorylation. So as a whole, if you look at this light dependent reaction, which is happening during the daytime especially, but light is not necessary here because there is no use of it. So in this, there are major 3 steps. One is carbon dioxide fixation here, carbon dioxide fixation, in which CO2 is required, and it combines with the RUBP, that is the one of the most abundant compounds, into GP. And the name of enzyme which is being used here, this is very important. The name of this enzyme is Ribisco. Ribisco is one of the most important enzyme here, and this is also one of the most abundant enzyme in the living organism. So this Ribisco enzyme is required to convert RUBP by combining with CO2 to GP. And there is optimum temperature for the C3 plants, that is 35 to 40 degrees. Above that, this Ribisco enzyme can do some reverse reactions also, just like a photorespiration. So, but in C4 plant, it is a different case that the optimum temperature is above 45 degrees centigrade. So there are many enzymes in the light dependent reaction like photolysis. Their enzymes are required, but names are not mentioned here in the CI syllabus or the A levels. So only this name, Rubisco, is included here. So this is the whole story about the Calvin cycle and the product of photosynthesis being produced here. That is glucose and amino acid and other organic compounds. So this is all about light dependent reaction. See you next. Thank you.